Hi everyone, my name is Natalia Córdoba and I am a biology student at the University of Oviedo in the north of Spain. Today I will be talking about assessing the prevalence of natural enemies on Pampas grass populations. Biological invasions are a major threat to global biodiversity and ecosystem function, with serious impacts on ecosystem services and human well-being. In recent years, the number of invasive species has grown exponentially, largely driven by human-mediated dispersal through global trade networks. The success of biological invasions is partly explained by the enemy release hypothesis, which proposes that invasive species face fewer natural enemies in their new environments. This reduction in pressure allows them to expand rapidly in both range and number. In their native habitats, these species are kept in check by natural enemies. However, once they are introduced elsewhere, they often escape these controls and proliferate more easily. In the case of invasive plants, insects are some of the most effective biological control agents, especially those that reduce seed production, which is crucial when the plant relies mainly on sexual reproduction. Without predators, invasive species may shift resources away from defense and toward reproduction, further boosting their spread. �々𝙧�10𝙧𝙀𝙀𝙀, commonly known as Pampas grass, presents gynodiasis, a reproductive system in which populations contain both female and hermaphroditic individuals. However, hermaphrodites produce very few viable seeds. As a result, the species behaves more like a dioecious plant. As an invasive plant, �々𝙧�10𝙧𝙀 outcompetes native vegetation, disrupts ecological processes, and causes significant visual and ecological impacts, particularly in early successional ecosystems. This species is a perennial grass native to temperate and subtropical regions of South America. It has become invasive in various parts of the world, including Europe, South Africa, Australia, New Zealand, and North America, where it was introduced for ornamental purposes, landscaping, and soil stabilization. Once established, it is difficult and costly to manage, requiring human and financial resources. One factor that may help limit its spread in non-native areas is the presence of natural enemies, which can influence its reproductive success and population dynamics. A potential natural enemy of �々𝙧𝙀𝙧𝙀 is 𝘨𝑛𝑜LEPIS cheloniae. This insect is a diptera that belongs to the 𝘴𝑒𝑐𝑖DOMIDAe family. It has been recently discovered in northwestern Spain. These insect larvae feed on the flower-developing ovaries, potentially decreasing the number of viable seeds. In the picture A, we can see a female adult of 𝘨𝑛𝑜LEPIS cheloniae, and in picture B, we can see the larvae. Our goal was to explore the biotic factors that might be limiting the regeneration of 𝘊𝙧𝙧𝙀𝙧. To do this, we examined several populations of the species across the range of environmental conditions, focusing on how the presence of natural enemies could affect the seed production. We surveyed 20 populations of Pampas grass across its current global distribution, including South America, North America, Europe, South Africa, Australia, and New Zealand. In each of the 5 regions, we selected 4 distant populations to ensure geographic variation. To evaluate how common flower predation is and how many seeds are actually available, we randomly selected 100 flowers from each plant. For each flower, we recorded whether it contained seeds, hosted natural enemies such as 𝘨𝙚𝙧𝙩𝙙𝙀𝙀, showed signs of fungal infections, or was empty, meaning either it wasn't fertilized or the seeds had already dispersed. Picture A shows a seed of cortaderia, and in picture B we can see the larvae of 𝘨𝙚𝙧𝙩𝙀. The picture D shows in the top a seed that is infected and below it one that is not. Our main hypothesis was that when the natural enemies are present, they could have a significant impact on the seed production, reducing the number of viable seeds per plant. We found incidents of flower infestation by 𝘨𝙚𝙧𝙩𝙀 larvae only in Europe. On average, 22.5% of the flowers in Europe were affected with larvae. This diptera is mainly found in European populations, particularly in Atlantic Europe, where its presence correlates with very low seed production. The majority of individuals were not affected with fungi, or the percentage was very low. However, fungal infections are more widespread than 𝘨𝙚𝙧𝙩𝙀, appearing in many regions and often occurring with high percentage of empty flowers. In this video, we can watch some of these insects and larvae in the flowers of Cortaderia celoana. Another potential candidate for biological control of Cortaderia celoana, based on surveys conducted in its native range, is this plant hopper. Further research is needed to assess if introducing population of this species could serve as effective biological control agents. Overall, describing this new species and its interaction with Cortaderia celoana is an important first step toward developing it as a potential biological control agent. However, further work is needed to fully assess its life cycle, geographic range, potential effects on non-target species, and whether it could be safely introduced to other populations of Cortaderia celoana across the world. This research was supported by the National Geographic Society and the Jardín Botánico de Gijón. We thank all the collaborators that helped with this project, especially Adrián Lázaro. Thank you all for your attention. Do you have any questions?